Good morning. What I wanted to teach you today was a subject that I found very important. It's very easy to understand, but something that you'll have to know as a scientist or researcher or a student. So the subject is cell culture. Cell culture began in the early 1900s. It was a method to study behavior of cells. They didn't know too much about how they grew or too much about the organisms that were responsible. So initially scientists used fragments of tissue, but gradually developed techniques to study the behavior of single cell and changed the name to cell culture. Well, what is cell culture? It is a process used to isolate or separate cells from a plant or an animal tissue and then place those cells in an environment where they can grow and thrive. Well, let's talk about the equipment that is required in order to do cell culture and research. Scientists culture cells in incubators typically 37 degrees Celsius with a 95% relative humidity and a 5% CO2. Primary cells that are isolated from humans and animal tissue using enzymatic trypsin or mechanical methods like homogenization or sonication. These cell lines can be finite or continuous. What is the function of an incubator? To maintain optimal temperature, moisture, sterile environment, and maintain optimal pH. First and foremost, sterile environment is very important. Many research projects will end with cellular contamination. It's important if you share an incubator with another scientist that y'all maintain it, communicate, and I would strongly suggest that you keep one cell on top and one cell on bottom and don't mix on shelves. So they're used to grow and maintain cell lines. So even though most incubators, as you see in the right this picture, they display the temperature and the CO2 and the oxygen on most, and it's automatic once you plug it in and have all the gas hooked up. But is that right? Is that temperature display, is that correct? And most of the time it's not, even though it's comes that way. And to calibrate your incubator, you will need a Firite analyzer. And it's this cool looking apparatus. And you plug up to your incubator, you pump the top 17 times, and where that liquid is, as you can see in the center, wherever that liquid lies, is your measurement of how much oxygen or CO2 that's in your incubator. So most of the time they're incorrect. So you have to find your manual and you have to recalibrate your machine yourself. And this is so important because when you're trying to troubleshoot and research, well, what happened? Did we euthanize the animal too soon? Did we use too much collagenase? Or did we not wash off the trypsin? What happened? So you're trying to troubleshoot. And I'm guilty of it myself, if you don't pay attention to this is where it ends and begins. If it's not healthy, then you will have disaster with your research. So there are two basic systems of growing cells. You've got adherent cells and you've got suspension cells. Adherent cells, the cells attach to a monolayer on the bottom of the flask. And the flasks, they call them treated flasks, and they're added with the cell culture medium, and they're left there for a few days to grow. But within that few days, you do want to change the media because the pH, what we were just talking about, the pH will change and they need fresh media. Suspension cultures, they're free-floating cells in culture medium and they have non-treated flasks, but you also have to change the media every other day in suspension cultures as well. It's just spinning it down and re-adding it to a new, fresh, sterile flask. So adherent cells, let's talk about this a little more. So cells like stem cells, fibroblasts, epithelial, lymphoblasts are types of adherent cells. Once you've got 90% confluency, they need to be passage. For cell removal, the chemical or mechanical methods can be used. So here's a little display I did today. So you've got your cell layer on top of the monolayer, and the flask is the bottom. So, and I left a break between the cells on purpose because you do not want overgrowth. If you got overgrowth, you've got problems and cells can change and it's not good. So mechanical method. The mechanical method involves using a spatula or a scraper to gently remove the cells off the monolayer on the bottom of the flask. However, there's a problem with this method, and it's inexpensive, but you have a risk of injuring the cells that have grown. So many scientists, including myself, we use the chemical method. So enzymatic method of cell dissociation. And this is when we use trypsin at a certain time or triple express. Triple express is preferred by many scientists. It is more gentle than trypsin. Both you have to wash off the cells, make sure you spin them down and put fresh media on them, or you will damage your cells. But after adherent cells are released, they will float around in the medium, and that's where you pipette them up and spin them down. So here is an example of cell passaging. So what I was just talking about, you've got your trypsin or your triple, and it breaks those cells off the monolayer. You pipette them up, put them in either 10 mil or 50 mil conical tube, spin it down, get a pellet. You resuspend after you remove the supernatant, and then you break them up. This only shows 2 petri discs. I recommend 4. Less cells on a flask or a petri dish is better. Lets them grow, have more space, so they can use up that nutrients in the media. So suspension cells is really what I work on in my research. They're single cells. They can be finite or continuous. Mine are finite, and they're much easier to passage. And here's an example of you've got your flask, you've got your media, and then you've got your cells that are floating around in that media. So that is the end of this introduction of cell culture. We will talk more about other things that can change how you culture your cell. If you have any questions, let me know. Thanks.